Welcome to this explainer. Look, in the world of pharma manufacturing, the stakes literally could not be higher, right? We're not just cranking out widgets, we're making life-saving medicines. And with that comes this massive responsibility to keep patients safe. Today, we're diving into a crucial and honestly sometimes misunderstood line of defense against a totally invisible threat, cross-contamination. We're going to unpack the science of MACO, basically the calculated limits that prove our equipment is actually safe to use from one batch to the next. Let's get into it.
So, here's our roadmap for today. We'll start with the cross-contamination risk, then define the MACO standard and understand the carryover concept. After that, we'll look at choosing the calculation method, exploring the worst-case scenario, and finally, how to avoid some really fatal calculation errors.
All right, kicking things off with Section 1, the cross-contamination risk.
So, is visually clean really clean enough? It's a pretty common sense assumption in everyday life, right? Like if a plate looks clean, it's clean. But in pharma, that assumption is incredibly dangerous. I mean, yes, a visual inspection is an absolutely crucial first step for sure, but on its own, totally inadequate. Active pharmaceutical ingredients are just incredibly potent. Traces that are literally invisible to the naked eye can still have profound, sometimes dangerous pharmacological effects on whoever takes the next product made in that same equipment. We absolutely need something way more rigorous than just human eyesight.
Which brings us to Section 2, defining the MACO standard.
Enter our defining metric, MACO, or Maximum Allowable Carryover. Basically, this is the absolute maximum quantity of residue from a previous product that can safely carry over into the next one without posing any risk to the patient. Think of it as the ultimate biological safety ceiling. But, and this is a huge but, I really want you to remember this. A MACO value is only as reliable as the toxicological data you use to justify it. You can't just guess or ballpark this number. It has to be grounded in current, highly accurate, scientifically sound tox assessments.
Why does this matter so much? Well, without these scientifically backed limits, we're basically flying blind. We literally wouldn't be able to tell if our cleaning treatments actually worked. MACO is the foundation here. It protects patients, sets the criteria for our analytical testing, and ultimately, it proves our compliance with good manufacturing practice, or GMP. Because, think about it. If an auditor walks onto your floor, they don't just want to hear that you cleaned the tank. No way. They want mathematical, scientific proof that the cleaning was sufficient. MACO is that proof.
Okay, moving right along to Section 3, understanding the carryover concept.
Picture a massive stainless steel mixing vessel in your plant. Yesterday, it was running product A. Let's say it's a super potent cardiovascular med. Then, the vessel gets cleaned. Today, you're using that exact same vessel to mix product B, maybe just a mild painkiller. The carryover concept is basically acknowledging the reality that even after a really rigorous cleaning protocol, microscopic traces of that heart med might still be clinging to the inside of the tank. And if they are, well, those traces are going straight into today's batch of painkillers.
And this leads to a massive paradigm shift in how we think about cleaning validation. The goal is not zero residue. The goal is a scientifically safe residue. Honestly, trying to prove absolute zero is practically impossible anyway. Our analytical instruments hit a wall at certain detection limits. So instead, the aim of cleaning validation is to knock those residues down to an acceptable, scientifically justified level where they pose exactly zero risk to the patient taking that next batch. Let's look at how we actually do the math in Section 4, choosing the calculation method.
I want you to think about the number 10 for a second. For the longest time in our industry, the 10 parts per million rule was just the undisputed gold standard. It was this blanket rule: no more than 10 ppm of yesterday's product can be in today's product. Just to put that into perspective, 1 part per million is like 1 inch in 16 miles. It's tiny, but here's the massive flaw. It's completely arbitrary. I mean, 10 ppm of a daily vitamin is wildly different from 10 ppm of a highly toxic oncology drug, right? So regulatory bodies are now pushing really hard for the industry to ditch this one-size-fits-all approach. Because of that flaw, we are firmly shifting away from those old arbitrary limits, like 10 ppm or just visibly clean, and moving toward health-based exposure limits, which you'll usually see abbreviated as HBEL, PDE, or ADE. This is the modern, rigorous standard. Instead of a lazy blanket rule, these health-based limits calculate a highly customized, safe exposure level based on actual clinical and toxicological data for that specific drug. It's a much smarter, patient-first approach.
But before you can even think about crunching the numbers, you have to gather your intelligence. It's kind of like a giant puzzle. First, you need process data, stuff like therapeutic doses and batch sizes. Second, equipment data. You have to calculate the total shared surface area. And third, the toxicological data, which includes the recovery factors for the specific swabs you're using. But a quick word of warning here. If your data gathering is sloppy or incomplete in these early stages, it leads directly to invalid limits. Basically, garbage in, garbage out, and your final calculation will be completely compromised.
Which brings us to Section 5, the worst-case scenario. Think of this as almost like a criminal profile for your most dangerous product. Look, in a facility making dozens of drugs, you aren't going to validate every single possible changeover sequence. That would take forever. Instead, you hunt for the worst-case scenario. You want the product with a super low permitted daily exposure, high pharmacological effectiveness, maybe terrible solubility, and a real stubborn tendency to stick to stainless steel. The logic is, if you can prove you can safely clean this absolute worst-case nightmare product, you scientifically justify that you can clean the easy ones. But pick the wrong worst-case product, and you end up with dangerous gaps in validation or limits that are literally impossible to meet.
All right, let's wrap up the process in Section 6, avoiding fatal calculation errors.
Okay, let's run through these common don'ts that auditors constantly catch. Things like using outdated PDE values just because someone didn't check for recent toxicological updates, or using the largest batch size instead of the smallest for the next product, which totally skews your concentration risk. How about ignoring swab recovery rates? Pretending your swab picked up 100% of the residue when in reality it only grabbed like 70%. And then, the math. Confounding units. Guys, a single typo mixing up milligrams and micrograms will throw your acceptance limit off by a factor of 1,000. Just one tiny unit conversion error and boom, your entire cleaning validation study is instantly invalidated.
And the traps don't stop there. It is incredibly risky to just copy and paste historical limits into a brand new protocol without actually verifying the assumptions behind them. Science evolves, right? Your limits have to evolve too. Also, you absolutely cannot just slap the same MACO value onto different pieces of equipment without assessing their unique surface areas. Oh, and if a product formulation changes or some new tox data drops, yeah, you have to recalculate that MACO.
So how do we avoid all this? We rely on good documentation practices. Everything you calculate must be completely traceable and verifiable. You've got to explicitly source and justify your tox data. You have to clearly show every single calculation and unit conversion. Seriously, show your math. Document the reasoning behind your batch sizes and surface areas. And finally, lock the whole thing down with signatures from a reviewer and an approver. That kind of traceable, verifiable evidence, that is your golden ticket to sailing through regulatory inspections.
I love this quote because it really hits the nail on the head. Establishing credible, compliant limits is not a solo sport. It requires a fully integrated, multidisciplinary team. Think about it. Validation engineers know the equipment. Toxicologists understand the biological risks, and analytical scientists know the testing limits. When those specialists actually collaborate, that's when your calculations become scientifically bulletproof and fully compliant with modern rigs.
So, to sum up our deep dive today, MACO is so much more than just a math formula on a spreadsheet. It is the vital, scientific link that connects a theoretical toxicological risk assessment directly to practical, boots-on-the-ground, cross-contamination safety on your manufacturing floor. It's the safety net. It's what ensures our cleaning processes are actually protecting the patients waiting for these medicines.
I want to leave you with this final thought. As the entire pharma industry makes this massive shift toward rigorous risk-based quality systems, is your facility truly prepared to scientifically defend its cleaning limits? Like, if an auditor asked today, could you trace every single calculation back to current, accurate data? Take these insights, really interrogate your own validation protocols, and make sure your numbers are building an absolute fortress around patient safety. Thanks for joining me on this explainer. Keep questioning, keep verifying, and keep learning.